I'll be talking about today is what I've been working on for the last 18 to 20 months as part of my KTP. That's sort of what I'm going to go through today. I won't talk more about myself since Massimo gave me such a good introduction, but I'll take the opportunity to just for those of you who don't know what a KTP is, KTP stands for Knowledge Transfer Partnership. It involves a company and a university partner, and they work together to foster collaboration for both benefit that would realize benefits for both the company and the university. So in this case, the company is Siemens Process Systems Engineering. It was PSE. Now it's SPSE, and the university is the University of Strathclyde, or more specifically, the CMAC Future Manufacturing Research Hub. So a little bit about Siemens PSE. It is an advanced process modeling platform, provides services and solutions for GPROM's formula products as well as other GPROM softwares. I was working with GPROM's formula products specifically. It is an equation-oriented technology. Those numbers are slightly outdated in terms of how many we are. We're just over 300 now, and it was acquired by Siemens about 3 years ago now. So in terms of the timeline, it was spun off out of Imperial College back in the 90s. That makes me feel quite old. And so it was since the 97 till about late 2010s. We were working, I say we, but they were working on getting the software up and running, commercializing it, making improvements to it, and after the acquisition of Siemens, now part of the core Siemens Digital Industries business. So the mission statement of the company is we bring next generation modeling and optimization technology to the process industry. This includes software, workflows, any other digital methodologies that are relevant to GPROMs, or for the pharma industry, GPROMs, formula products. So helping bring high quality drugs to patients faster is well, basically what we do. How this is done is through R&D efficiency, using fewer experiments, saving time, material, and effort in the labs to get more robust methods to generate larger quantities of API, reducing the risks, again, by using softwares, and allowing for a tech transfer from upscaling or scaling from batch to continuous. Using engineering, so I mentioned that, scaling up or tech transfer, moving from small scale to a larger scale, or batch to continuous processing, and allowing for monitoring and soft sensing using the platform. So this is what the PSE ecosystem looks like. Let me see if I can get a pointer. You see that on the screen? Yeah. So perfect. So PSE or SPSE has a lot of moving parts. You've got the alliance with the pharma companies. You've got the systems-based pharmaceutical vision. And then you've got the funded opportunities and the collaborations with CMAC and other university partners. This helps develop GPROM's formulated products, GPROM's process, model builder, and so on. And with the Siemens acquisition, this has just made things go faster and brought more expertise in the same field. So a little bit about the project that I have been working with as a KTP. So in terms of the aims of the specific project, I'm working to make it easy to model on GPROM's formulated products. This may be with including example cases in releases, robustness improvements, inclusion of workflows, etc. So you don't have to read all of that. It just says basically that I'm working to make your life easy when it comes to modeling. So in terms of the model libraries, this again is slightly outdated. There's a couple of new libraries that have been introduced and another one that is upcoming, which I will talk about in a little bit. But this is sort of the breadth of the libraries that are available in GPROM's formulated products, all the way from active ingredient manufacturing all the way to drug product performance and also food manufacturing. As part of the KTP, I've worked on a few of these, not all of them, of course, and there's a couple others. There's one more that I'm also working on. Again, I will talk about it a little bit in the outputs. In terms of the outputs, I will be talking a little bit more higher level, so I'm not going into details of what the models are, the kinetics behind the models. I'm just talking about how the project has fostered innovation and collaboration between the two, the industry and the university. So one of the things that has been developed through the project are digital design workflows. So these are guided workflows that will help you model processes using specific model libraries and gPROMS formulated products. So for now, these have been developed for crystallization, seeded cooling, and pressure filtration libraries. You can see it looks quite intimidating when you look at it. We are working to make it smaller so that you're looking at what you really need to see and not everything else. But again, the idea is that if you start from phase, So if you start from phase X or phase 0 on that side and move all the way to the end, the idea would be you would have a robust, optimized process for crystallization of your chosen API. More specifically, so if you look at one of the phases, it looks at how to configure a simple model. So it will take you through how you define your material system, how you define your crystallization equipment and operations, and move on to running it and seeing that it runs. And there's loops to try and troubleshoot why it's not running. Now this is also being developed into a training material that will go on to a self-paced learning platform. Maybe soon enough, hopefully. I'm working on that, so it's basically as fast as I can do it. Another thing that I have been working on is developing help documentation for guiding model validation in gPROMS formulated products. One of the things that you need to do in gPROMS formulated products, if you're not familiar with, is when you're trying to estimate your kinetic parameters, you need to set up a model validation entity, specify initial guesses and bounds on how to do that. So the idea through this KTP was to guide that for the pressure filtration unit and define how you would choose your initial guesses and bounds and, well, help you do your model validation or kinetic parameter estimation a lot easier. So this has already been implemented. I know it says ongoing because it keeps being refined, but this is there in the help documentation within gPROMS formulated products, where I know the image is not very clear, but you can see that based on the kind of kinetics that you want to estimate, whether it's cake resistance or specific cake resistance, formulas like the Cameron-Kazeni cake resistance equation or Endo-Alonso and so on, these, oh, thank you for the mood lighting. So you could go onto the help documentation, and that would be a good way, a good guide at least, we hope, to help you get there, get your validated pressure filter model. So again, this is not only for pressure filtration. There are others available within the help documentation, but through the KTP and through the collaboration, I was able to develop this for the pressure filtration unit. Another thing that was developed through this was a continuous filtration case. Now, this was done using data generated at CMAC by other researchers, so Sara Ottoboni, who you heard from yesterday. And this was data collected using the AWL continuous filtration unit. And the idea was to try and mimic this in gPROMS formulated products, basically. And the model that was used to do this was the Cameron-Kazeni cake resistance model. See, what you would normally do is start with a batch pressure filtration unit and then connect everything else and translate it into a continuous pressure filtration unit. The idea here was that the work would make its way into an upcoming release. The work was also presented at Philtech a couple of months ago, where the optimization of the way it was done in the lab was conducted well through gPROMS, and I was able to realize a significant reduction in the time taken to do this process. Now, in reality, you wouldn't be able to minimize the whole 1100 seconds or about 15 minutes of time. But the idea is that through gPROMS, you're able to realize this, and you can see that there are parts where you can make these improvements. And I'm happy to speak to you about this outside of this, or if there is time after the talk, I'm happy to answer any questions. Another project that I've been working on as part of the wider hub mefenamic case is the wet milling experiments, or wet milling for seed generation of mefenamic acid. Now, this will form part of an integrated systems model. And the idea is that, there were experiments done previously that were used to generate a model, and they were used to guide further experimental work based on the findings from a design space exploration, which I haven't shown here. But the idea is that based on the findings from the model, someone from CMAC was able to go back into the lab, get more experiments, and now those experiments are being used to revalidate the model. So it's more model-driven experiments, model-based design of experiments, and this work is a poster over there in conference room 4. So if you have any questions about this, I'm happy to answer them. It will also be presented at a conference in September in Madrid, hopefully. Another thing, so I mentioned a new model library that's being developed within GPROM's formulated products is for an agitated filter dryer or enhanced dryer. So the development is done within GPROMs. The idea was, can we collect some lab data from CMAC or design some experiments to guide the model testing and model development? So just last month, I came down to CMAC and worked with Sarah and Chris Price from the chemical engineering department to gather some filtration and agitated drying data using the AWL unit that is based in ChemEng, or the newer version that you may have seen in the lab tours yesterday or today if you go. So the model is currently in development. You can see sort of an example flow sheet that I created just a couple of weeks ago. And those lab experiments were done using, as I said, the drying rig that is in the AWL drying rig in ChemEng. Again, the idea was to collect the data on moisture content development or moisture content evolution over time and use that to analyze the model and test it. Another key output of this, so you've seen sort of the benefits that have been realized by the industry, so by PSCs, in terms of model development, robustness improvements, example cases. So what's in it for the university? So lots of conference presentations in the last year and a half. Yeah, COVID limiting, because most of them were virtual, but I ended up presenting was involved in about 3 oral presentations and 3 posters at various conferences in the last 6 months, really. And these ranged from simple model configuration, a tech transfer from batch to continuous, or if you start from things like, if you look at using SAFT gamma me to drive your solubility predictions and so on. So things like this are benefits that the university would realize, and also there are some publications that will come out of some of these conferences as well, or some of these outputs. So benefits of the partnership for me. So the collaborations with the Siemens PSC in general. So I've been part of the active ingredient manufacturing practice area, the product development team, where I've worked on improvements of the model libraries as well as example cases and model enhancements. So I already mentioned the continuous pressure filtration case, but there is another one on the centrifugal filtration case that I'm working on as a tech transfer from pressure filtration to centrifugal one. In terms of knowledge transfer, I did mention the workflows that will be translated into an on-demand knowledge transfer platform or a self-paced learning platform. So in terms of that, it's not just the workflows, but looking at other developments that we can make to make modeling easier on GPROM's formulated products. And finally, trying to develop a new case for a new model library on hot melt extrusion. And I'm working with the formulation manufacturing team on this as well. With CMAC, so you heard from Sarah yesterday on her filtration work that she did for the hub. We're trying to extend that a little bit more to include impurity tracking in the wash stages and a little bit in the filtration stages. So part of this work has been presented at Filtech early in March, and more of it will be presented at the World Filtration Congress in October. You can speak to Sarah as well. She's in conference room 4 and 5. She'll be happy to talk about this to no end. I'm also working with Bilal, who you heard from yesterday on spherical agglomeration, but I'm working with him on a wet milling side of things, trying to validate a crystallizer with a wet mill and recycle. This, again, has been accepted for a conference a conference presentation in Madrid. Could be a potential publication and may also become a model in an example case in an upcoming release. And I'm happy to speak to you about this in the same conference room. And for the wider hub, trying to get together a systems or an integrated end-to-end model for the production of mefenamic acid. And the output of this is still to be confirmed. And again, it's still work in progress. So let's see where that goes. So thanks for your attention, and I'm happy to take any questions.
Hi, good morning, everyone. Awesome to be here. Awesome to see people in the physical realm and reconnect with friends. I'm sure everyone has said this at the start of their presentation. I'd like to do three things this morning. Give you a little bit of an introduction to Wyoming, because Siemens was mentioned on the previous presentation, and I don't think there's anyone who has not heard of Siemens, but can probably count on the fingers of one foot who's heard of Wyoming. So I'm going to try and put that right. I'm also going to talk a little bit about collaborations, specifically from an SME perspective, and try and figure out if it's a good idea to engage SMEs, because we don't have the scale of Siemens. But spoiler alert, it is a good idea. I'm going to hopefully show you that. And then I'd like to give an example of an active collaboration that we're working on with CMAC, which is around a platform called LabForce AI, which I think is just an awesome example of a great collaboration, things coming together. So Massimo mentioned work in life science. We're a team of about 50 people. We work across manufacturing, research, supply chain. We also work with instrument manufacturers. We work with some diagnostics, clinical trials, a range of topics within life science. But one thing that runs through everything that we do is managing data and presenting data. We're not from there. Last night at dinner, I think I bored people with the origin story for the name Wyoming. I'm not going to do that here. My apologies to my table colleagues last night. We're not from anywhere in America, although the United States is our biggest market. About 70% of our work is for clients based in America, but we are entirely based here in Scotland. About 50 miles from here actually I don't know what direction. I've kind of lost my sense of north, south, east, west, but we're in Edinburgh. What do we kind of do for life science organizations, CMOs, CROs, prime manufacturers? We provide a range of services, customer research, planning. We build applications, we work with data, we design products and services, and we maintain them. But I think the thing that we're really good at is expressing the right information for the right person at the right time. It's a kind of combination of all of those skills. We try to spend a lot of time on research, understanding the audience. When you design a tool, a workflow, a method, trying to think about the interactions that the audience, the intended user base, will have with that tool. Different levels of expertise, requirements for different outputs. And we try and think a lot about those kind of issues, map them out, and try and design for the people who are using the applications. So we use design thinking a lot in our day-to-day work. I'm actually here with one of my colleagues, Sophie, who's over there. So if you want to know a little bit more about design thinking and how we apply that, Sophie is here with me today. One of the things that we're good at as an SME, a group of about 50 people, is we move fast. It's one of the great things about SMEs, agility. And we try not to spend 2 years trying to get something precisely right, but try to, in some parlance, fail fast or learn fast, kind of test, measure, learn, and quickly iterate to high quality outputs. So focusing less on being right, but being really good at being wrong and moving on, using analytics smartly to get to a better place. So a little bit about collaboration. When we started as an organization 15 years ago, I think it's fair to say, like any SME, we would have pretty much done anything for anyone, and it's all about establishing your place in the marketplace and staying alive. But now that we're a bit more of an established business and we've established good relationships with long-standing relationships with instrument manufacturers, CMOs, CROs, etc., we've got a little bit more flexibility to change our model and thinking more in terms of long-term and how do we get kind of interesting projects for our teams, not just for the next quarter or the next couple of quarters, but for next year, the year after, things like that. And that allows us to enter into collaborations which perhaps don't have an immediate commercial endpoint, and we can invest a little bit from our perspective. And it also makes for really interesting work to get involved with. As an SME, so if I just step back, last night as an example, at the dinner, I was sat between a representative from Pfizer and a representative from AstraZeneca, and it's really challenging for SMEs to be able to gain access to those kinds of organizations and to understand what's top of mind for those organizations, what are the points of pain that those organizations are facing, what are they trying to solve for. So there's enormous value in collaborations and being involved with organizations like CMAC. We gain visibility as an SME that we would struggle to find, and we inherit some authority from the community that we're part of, which is enormously valuable. Of course, you've got to follow up, and you've got to actually be good at something. You've got to be able to deliver and contribute, and I think we've got a great track record of doing that. But collaboration is great for SMEs. On the other side, what do you get from an SME? And I recognize that I should have reordered this slide, because the last thing I mentioned is the key thing that should have been top of the list. But aside from my editing skills on PowerPoint, the specialist expertise, we are focused on a very narrow range of topics. We get very good at being in a small area of expertise. And that's one of the great things about SMEs, the specialism, lots of knowledgeable people focused on one area. But you also get simplified processes and approvals. You don't have to go through 17 vice presidents of procurement to get something done. You can speak to decision makers. You can get action fast with SMEs, and we try and move with pace and agility. My experience of trying to initiate partnerships is that you have to persevere. You have to try a large number of things, and some of them stick and some of them are good. We've been involved with some really interesting collaborations that unfortunately didn't go anywhere. One really interesting one about wearable sensors that we thought was a great idea, and the partnership just didn't go anywhere. So you sometimes, it's not always the direction that you think things are going to go that is successful. So continuing to adapt and pivot. You've got to give to get. As an SME, you've got to demonstrate some value first, I think, before the partners, engage fully. And doing things like showing some of your skills, participating, perhaps doing some demonstrators, POCs, things like that, are good ways to demonstrate your value. And being fast to value, not waiting 2 years to surface something, but being able to react fast and give something of value very, very quickly. I think we're great at doing that. We're very focused on chunking up big projects into deliverables and making it happen. To try and determine a good fit for a partnership, you kind of want a degree of overlap between the partners, but you don't want to be, almost completely overlapping. You want to have some separation so that there's a contribution, an obvious contribution area for each organization. And for SMEs, I think finding your thing is a key part of making partnerships work. We say, if someone asks for a list of services, we say, yeah, we do CX, customer experience research, we do user experience design, we do data visualization. Those are things that we have as notional services. But what we're really good at, the one thing that we say that we're really good at, is showing the right data to the right people at the right time. And it uses all of our skills in combination, and it's a really powerful way of getting smart applications that have got quick uptake, work at scale, and are easy to service and maintain. So that's our thing, and that's what we bring to partnerships. So an example, so LabForce AI. I have to say that this is a great partnership, and not just because I'm standing here in CMAC, but it is truly a great partnership. I've really enjoyed working with the team at CMAC. It's been difficult over the last couple of years because of what we all know, but great to reconnect with people in the physical realm. What is LabForce? Well, there's an AI. So I believe it's room 6, poster number 20. Tony, I don't know if Tony was... Tony up at the back. In room 6, poster number 20, I would encourage you to have a look at that, because it goes into detail behind the algorithmic solution. What our role is creating a platform for smart algorithm, some very smart work with data, putting it onto a platform and sharing it widely, making it work at scale. And that leverages the respective skills of both organizations, the deep understanding of chemical processes and AI, being able to bring that, and then having a partner that can provide software at scale on a platform that delivers security, subscriber management, education, all things that you need in a platform. It relates to solubility. I am not an expert on solubility, so I am going to step away from any questions and defer to experts in the room. But one of the things that we're trying to do is support continual improvement so that there's multiple releases of this, and using community data. So benefiting from people who are doing experiments, pulling all of that data so the AI gets better and better, and the efficacy is shared with everyone who is pulling data. I think it's a great model. Single platform that will support multiple apps. Solubility is the first one that will be coming out of the gate. We want to gain data scale from working with partners, pulling the data, and I've heard FAIR fair data mentioned a lot, and the challenges with particularly the I of fair, interoperable, common issue with fair data. But this application really does speak to the interoperable and reusable elements of data. And again, poster number 20. I encourage you to look at the QR code, which will bring you to a landing page that shows some information about the platform. We have a video running in the exhibition area. Sophie and I will be there later if you want to chat. Some of the things that we're going to bring to the benefit of audience: freeing up lab equipment, quicker, more accurate results, great customer benefits, tangible benefits that will save time in the lab. So I encourage you to do that. I might give you a little reminder about the QR code. So very quickly, in summary, collaborating with SMEs, specialist knowledge, pace and agility. We can move fast, we can get things happen, and you get direct access to decision makers. And for SMEs, partnerships are great because we get to learn about the things that are points of pain, big ticket issues for large organizations. So we love collaborations and keen to do more. And just in case you didn't catch the QR code first time round, I thought I'd just drop on a couple of memories. So thank you very much.
Thank you, Rob.
In this presentation, I will focus on how an academic idea on innovation in pharmaceutical manufacturing can lead to a spin-off company in which this new technology is further commercialized. So in this presentation, I will give you a very brief introduction to pharmaceutical freeze drying for those that are not familiar with it. Then I will focus on the first part, the academic work that has been done since 2010. Then in the second part, I will focus on the valorization trajectory through the spin-off company ReaVita, which was established in 2018. I will also tell you a little bit on the current status, but also on the future steps and perspectives. So large molecules are considered the key driver of growth in the pharma industry. And when you look at the biopharmaceutical products that are on the market today, almost 50% of them are in a freeze-dried form on the market. So this clearly shows that freeze drying is a very important and a preferred way of stabilizing biopharmaceutical products. The freeze drying process itself, and now I will start a little bit from my academic background and also present you a little bit the academic steps that we've been taking. So the freeze drying process itself consists of 3 consecutive process steps. It exists with a freezing step where the aqueous solution with the biopharmaceutical is converted into ice, and the solids themselves, they are crystallized or transformed into an amorphous system. Then in the next step, the system in which you have these vials is pulled under vacuum, inducing ice sublimation. And then finally, there's also a secondary drying step where the unfrozen water is removed by desorption, because during freezing, not all the water is converted into ice. Some of the water is dissolved in the amorphous matrix, for instance. You can also see here at the bottom that the freeze drying process itself is quite a time-consuming process. Freezing up to 12 hours, ice sublimation can take several days. Desorption can also take several days. So freeze drying processes of several days are not uncommon, even a week. And here you see the conventional freeze drying equipment, which is used for decades. So it's batch equipment. The system is let's say, filled with tens of thousands of vials on temperature-controlled shelves, as you can see here. So after the loading of the system, the door is closed. The freezing process starts. After freezing, the whole system is pulled under vacuum, and then this induces primary and secondary drying. Actually, my freeze drying research activities started already during my PhD and postdoc period back from 2007 to 2010. And this, when I was at the start of my PhD, 2007, that was just a couple of years after let's say, the PET guidance was published in 2004. My PhD promoter at that time told me, okay, now you have to develop PET solutions for conventional batch freeze-drying processes. So let's say I had to work a little bit on how to move from a fixed process recipe without a lot of process monitoring and control to, let's say, a controlled freeze-drying process. So, of course, a lot of academic research was performed. You can see these pictures here, which are typically pictures that you can take in an academic research group where you try to implement sensors, where based on these sensors you try to collect or to try to develop process understanding. You try to implement sensors that allow you to visualize how the product is behaving during the process and so on. But towards the end of my postdoc period, we came to the conclusion that there is quite, let's say, some conflict between the goals which are stated in the PET guideline and conventional batch freeze-drying. It was quite challenging to have, let's say, a process monitoring and control system implemented that allows you to monitoring and control the process at an individual product level. Taking this into account, there are also some inherent, let's say, issues or challenges with conventional batch freeze-drying. There is a high production cost, a huge footprint, high capex and opex cost. It's a very slow process, as mentioned. Upskilling is also not very straightforward. When you start developing your batch freeze-drying process in a small lab-scale freeze-dryer, and then you need to scale it up to a production-scale freeze-dryer, that almost requires complete re-optimization and revalidation. But also from a quality perspective, there are some issues. And let me first explain what the potential issues are. So here at the bottom of the slide, this is a reference from University of Eastern Finland. You see actually the top view of a shelf. So on these shelves, you have some hundreds to thousands of vials. So every small circle on this figure here is representing an individual vial. And the color is referring to the process progress. So what you clearly see here is depending on where the individual products are in the batch freeze dryer, they undergo a different process. And this is, of course, quite in conflict with, let's say, what process monitoring, what process analytical technology stands for. And a couple of years ago, for instance, a company like Pfizer, they presented on lyophilization issues at the PDA conference, and this was also what they mentioned there. There is, let's say, an inherent quality potential problem with batch freeze drying. Also, when you have tens of thousands of vials in such a batch freeze dryer, the impact of batch loss, defects, and maintenance on supply chain is huge. There is also clearly a lack of flexibility. However, when you think about the processes before and after freeze drying, well, filling of the individual vials, that's a continuous process. Capping, packaging of these vials, that's also a continuous process. So it would be great if there is a continuous alternative for this. Furthermore, when we also think from a product-related perspective, there is also a new need. Personalized medicines, they need flexible production campaigns and supply. There is also an increased importance for low volume and high value products, so this also requires flexible manufacturing technologies. But also new products, several new products require better controlled process settings. And then at the start of my professorship in 2011, I had a colleague with whom I was working, and we had a new freeze-drying ID, a freeze-drying ID in which process and understanding is crucial, where we also try to process monitoring, but also control at an individual vial, at an individual product level. So not only the monitoring, but also use in real time the information that you get from your sensors to guide the process towards the desired state. This creates an inherent real-time release potential. It's a continuous process, but it's also a product enabler. And this technology is now, let's say, covered by several patents. In the first movie, you will again see from an academic perspective what the new technology stands for. So you will see we apply spin freezing. So freezing of the product is a little bit different. So you see we use a very simple setup. And then you can see that cold gas is used to cool and freeze the solution. It's by the combination of gas flow and gas temperature that you can perfectly, let's say, determine or perfectly set the cooling and the freezing rate. But at the same time, as mentioned, process monitoring and control is crucial. Product temperature is one of the most critical process parameters during freeze drying. Well, when you apply a continuous approach, well, the vials, one after the other, as you will see in a moment, are passing in a line through the continuous freeze dryer. So the vials are kind of naked. You have full access to the vials by means of sensors. And in this case, there is the opportunity to use a contactless temperature measurement system. So this is thermal imaging. And based on this thermal imaging, for all the vials that are processed, you get constant temperature information during the entire freeze drying process. Here at this, let's say in this status of the movie, it's during the freezing step, but also during primary and secondary drying, product temperature is crucial, and this information is collected. The information is not only collected, it's also used to give feedback control to the process settings, to make sure that your individual products, that they have the desired, let's say, process temperature profiles during the entire project. I'll let the movie continue, but what I also want to say here is that based on this technology, we were also able to do the process much faster. Freezing can be done in a few minutes. So in a batch freeze dryer, you can, let's say, cool between 0.5 and 2 degrees Celsius per minute. Well, here you can do it between 0.5 and up to 80, 90 degrees Celsius per minute. So you have a much wider cooling rate potential, and this can also be crucial for certain types of products. For instance, cell-based products, they really need a dedicated cooling freezing trajectory. Then in the next step, well, ice sublimation can be done much faster. You can see it in 30 to 60 minutes. Due to the fact that we do spin freezing, well, you have a thin layer at the sidewall of the vial. So you have a much thinner layer and you have a larger surface area. That allows you to do the ice sublimation, but also the desorption much faster. So let's say the freeze drying process now is reduced from days to 1 to 2 hours. This movie is still progressing at the vial level. So here you see what happens at the vial level. Now the freezing is done. As you will see in a moment, for sublimation and for desorption, you need to supply some energy. Well, we supply energy to radiation. You will see that in a moment. So now the frozen vial will be slowly rotating in front of a radiator. This radiator is placed outside the freeze dryer. But again, at the same time, the product temperature during sublimation and desorption is crucial. So this vial is again continuously monitored with a thermal imaging system, and that's giving feedback control to that radiator to make sure that you don't supply too much energy, but that you supply sufficient energy to dry as fast as possible. And this is then how a final continuously freeze-dried product looks like. This was a movie at let's say, a vial level. Of course, you need a certain production capacity as well. That's what the next movie will show you. Try to restart it. So what you will see in a moment, and that's here, the vials, they immediately come from the filling lines. So through the filling lines, they are led through a spin freezing module. Next to the spin freezing module, there is a sublimation module, and then there is a desorption module. So it's a modular system. One remark, between the modules we have load locks, because for instance, spin freezing is performed under atmospheric pressure. Primary drying, secondary drying is under vacuum, so you need a fast way to transfer the vials there. That's based through load lock systems, and you can do that in seconds, also in a clean way. Scale up is also, let's say, flexible. It's a modular system, so based on the size of the modules, but also by placing modules in parallel, you can, let's say, create the production capacity that you need. So this was all, let's say, research done at Ghent University. Well, then, in the final step of the Ghent University research program, we obtained funding to build a first demonstrator prototype. So this was the first demonstrator prototype continuous freeze dryer which was built. As you can see, it's of course a non-GMP system. However, GMP principles were taken into account already during the development. So what you have here is a manual infeed of the vials. Here you have a load lock system. On the left, there was then the spin freezing module, and then on the right, you have a drying module, the vials coming out at the same way. What I also want to mention is that we used a model-based approach, a model-based design approach. Before building this prototype, we first physically described in mechanistic models what the process should do with the vials. And then we used this knowledge to build the equipment around that. So model-based design approach was used. But since we now have the models in place, well, this also provides us a software opportunity. So we have now the models that we have anyway built, implemented in user-friendly software. So if you have a new product, well, based on the product properties, you can fill that in the software, and then the software will tell you how you optimally set the continuous freeze-drying process conditions. So with a very few amount of experiments, you can, let's say, immediately optimize your formulation, but also your process. Then here, as a final movie on this slide, here you can also see an example on the feedback process control, which is inherently implemented here. So as mentioned, the vials, they are continuously monitored with thermal imaging system, where that information is in real time, and this is an example for primary and secondary drying, giving feedback to the radiators. So here you can see we need to control the temperature of the product during the drying phases. The product temperatures may not exceed a certain critical temperature. Well, you can see that this information is, let's say, immediately used to set the radiators, which are the red line. So it's constantly steered, this process. And that was actually the end of the academic phase. So once this prototype was ready, once the control systems were implemented, well, that was the start of the spin-off company from Ghent University, Reavita. Also for myself, it was a certain change. I stepped back for 50% at Ghent University and also got the opportunity to join this company. Of course, the first step there was to establish a company team and also a company organization, so totally different focus. Immediately at the start, since we had that prototype available, we had the opportunity to start and test and evaluate the technology with several pharmaceutical companies. So at this moment, and we're still doing that, the technology is tested for a wide range of different pharmaceutical formulations, large molecules, vaccines, cell-based formulations, but also pre-filled syringes, dual-chamber cartridges, highly concentrated protein formulations, mRNA, LNP-based formulations. They also need freeze drying, as you may guess. So that's what we immediately started. But at the same time, we also had to further develop the technology, because at the start we had a non-GMP demonstrator, but it's clear for pharmaceutical production, we need to be able to produce, let's say, under GMP in the future. Actually, two technologies were developed then by the company, a small R&D unit, I'll come back on that in a moment, but also a GMP-ready production scale equipment was developed. Furthermore, the software was also further developed, further fine-tuned. Let me first move to the R&D tool. We call that the SVU. That's a single vial unit. So in this system, you simply process one vial. So you do the processing of one vial. You do that in the early phases of research and development. So as I mentioned, when you have a new product that needs freeze drying, well, based on the formulation properties, based on the vial dimensions, based on the filling volumes, well, with the user-friendly software, you can immediately determine what the optimal freeze-drying conditions are, and you can immediately execute that in this single vial unit. Also need to emphasize that the sensors and the advanced process control systems are also implemented in the single vial unit. So with a very low number of experiments, with very limited material consumption, you can do the entire formulation and process development. You can do, for instance, very fast DOEs. When you have optimized your process in this single vial unit, well, the optimized process for your product can be one-on-one transferred to the production scale equipment. Because what you do here is, the process signature that your vial experiences during the process, that can be, let's say, replicated in the continuous freeze-drying equipment, thanks to the sensors and the advanced process control system that you have implemented. So that was a first development. These single vial units are now sold worldwide. And then in the next step, we also had to develop a GMP flex equipment. Why do we call it GMP flex? Well, because it's a modular system, and flex refers to the flexible production capacity. So depending on the capacity that you need, well, you can use larger modules or you can use more modules. As mentioned, it's a very fast process. Well, PAT is crucial there. Every vial is monitored and controlled. So this creates a real-time release potential. In this movie, you will see, we call that a vial's way. Well, you can see how such an individual vial, what the vial's way is through these GMP Flex continuous freeze-drying equipment. That's actually almost my final slide, just to give you an idea what the final developments at the company level are at this moment. So you see the individual vials immediately coming from the filling lines, so the system can be coupled to a filling line. You don't need the large clean rooms anymore for loading and unloading of batch freeze dryers. You simply connect it directly to filling lines. And as you can see here, well, all the vials, they are continuously monitored, controlled, and based on load log systems brought from one module to the other. What I can give as a final remark here, well, it's also an enabler for certain types of product. As mentioned, you have a much wider range of process settings during freezing. Can be very important for freeze drying. Since we can process at very low pressures and also without condensers, that's just a detail, we also have the opportunity to process products with very low critical temperatures in a very fast way. But also, due to the fact that you have a thin layer at the sidewall of the vial, the reconstitution time can be much faster. We've seen that this is very important for certain types of, for instance, highly concentrated protein formulations. So during the movie, you could see the vial's way through the GMP Flex system up to stop ring. Now, in the final shot, well, you see how such equipment looks like. But as mentioned, scaling up, well, we don't talk about scale up, we number up, we scale out. So this is how this is done. So the current status is this equipment. First equipment sold in the US, EU, and Japan. We have also entered pre-engineering phases for the GMP Flex system with the customers. But at the same time, we also provide services. We don't want to sell stainless steel. No, we want to provide solutions for the products of our customers. So we provide also services for formulation and development linked to this technology. So what are the next steps? Well, we want to have this system, of course, in a pharmaceutical company for manufacturing of product. And I think a very important next milestone for us would be the realization of a first clinical batch, the use of the technology for products that are injected in patients. And also from an organization perspective, further expansion of the Vita team and organization is also very important. It's clear that we need a lot of engineering profiles, and finding engineers, that's quite a job at the moment. So with that, I hope I was able to give you a presentation, an introduction on how a new manufacturing technology started from early R&D phases in a university up to a spin-off company. Thank you very much for your attention.